So today I'm going to be talking about speciation of milk snakes and specifically how we can delimit species and then use those to infer a species tree for Lampropeltis. So one of the things we want to have when we make a phylogeny is all the extant taxa. And so for any group you're looking at, including all extant taxa, is extremely important because when we have missing taxa, we're going to have a less accurate phylogeny. This is going to be with respect to both the topology, which is somewhat obvious because if you don't have all the taxa, you're not going to know who's sister to who necessarily. But also maybe a little less obvious is node density. And so when we have missing taxa in our tree, we're going to have more nodes that are further back in time relative to younger divergence times. And this may signal early diversification, so downstream analyses may be misinterpreted because of this. So just to show an example of what I mean here, let's say we have this phylogeny with 50 taxa, and if we take a look at the node density on the y-axis and the branching times along the x-axis, we see that most nodes are clustered around 3 million years ago. But if we take the same phylogeny, prune that down to only 25 taxa, we see that that node density shifts to an older overall divergence time of about 5 million years. And so compounding the problem of estimating divergence time with missing taxa is the use of gene trees instead of species trees. So everybody probably knows now that we want to use a species tree approach whenever possible because of issues such as incomplete lineage sorting. But in addition to incomplete lineage sorting, gene trees are also going to overestimate divergence times. And this is because genes are going to diverge prior to species. All the genetic diversity is already present by the time the species diverge. And in addition to that, gene tree methods also don't take this into account. But we have multi-species coalescent methods that we can use that explicitly estimate species trees and take all the gene histories into account. Hopefully you're going to get more accurate divergence times that are likely younger when you use a method like this. And so if we're going to make a species tree, we want to not use a gene tree and we want to include all taxa, the first thing we might have to do for some groups is delimit species. So traditionally we've done this based on morphology, but we have the issue of cryptic taxa, which we know for a lot of wide-ranging species is often quite common. And so a lot of phylogenetic and phylogeographic studies describe species and lineages based on patterns that you get from a tree. So a good example of this is Bourbon 2002 corn snakes. So we have this wide-ranging corn snake species, and it turns out that there's actually 3 distinct lineages in there, each with their own separate evolutionary trajectory. But even though this is valid and is often found to be robust and more data can be added that corroborates this, it's not an explicit test for a proposed species hypothesis. And in science, we're always trying to really test an explicit hypothesis. So one way we can do that now is using programs such as Bayesian Phylogenetics and Phylogeography, which I'll refer to as BPP throughout the rest of my talk. And what this program is able to do is it's a coalescent method that you can use to verify a species hypothesis, and it gives you a posterior probability support. So you've got some sort of statistical support value that goes along with whether you can you can find species distinct that you're proposing. One thing to know about this method is that it assumes no or low migration between species. So in some ways it's sort of similar to the biological species concept in that you don't really have much gene flow, but you could have a little. And another thing is that there's a user input guide tree, and this is really important for using this program BPP because you need to propose what the species are in this guide tree and also what their topological relationships are to each other. You can do this by getting a gene tree, so if you have a mitochondrial gene tree and you want to test are those species delimitable, you can use a population structure analysis, something like Structurama, or you can use something based on morphology, like subspecies designations. And to show an example of how this might work, let's say I have 3 species of cobra right here in this guide tree. So I'm saying I think there's 3 species, A, B, and C. I think these are their relationships. You would run BPP on this, and you would get some sort of results. They might look like this, where C is considered distinct with 100% posterior probability support here. But A and B, that node is collapsed. It can't distinguish between the 2 as separate species. So that brings us to the group that I'm sure we're all here to really see today, not just talk about gene trees and delimitation, but milk snakes. So Lampropeltis triangulum is a member of the New World snake tribe Lampropeltini, and I'm sure everyone here is familiar with these guys. So these are the rat snakes, the corn snakes, the milk snakes, the king snakes. And what's really cool about this group is that there's been a lot of phylogeographic studies done. A lot of them are really wide-ranging, and there's a lot of cryptic diversity. But the most wide-ranging of all these snakes is the milk snake, and no one's looked at this in a phylogeographic context. So it's among the widest ranging of New World squamates, and not only is it wide-ranging, but it's also got these 25 subspecies with incredibly diverse body sizes, habitats, and ecologies. And if we take a look at how this would look with their distribution and some of the variation in color patterns for the subspecies, they range across the US, throughout all of Mexico, into Central America, and going all the way down into Ecuador, Colombia, and Venezuela. And you can tell that just from this range, they're going through a lot of different diverse habitats. They've got this diverse color pattern. And just based on this, we might think that there's cryptic diversity, or maybe not even so cryptic. And so the goals of this are, first, let's estimate diversity within Lampropeltis triangulum and figure out what's really going on here. Infer a species trait for the genus. And then we also want to look at the timing of diversification. So previous studies have indicated that Lampropeltis diversified only during the Miocene and Pliocene. And this is somewhat counter to a lot of temperate North American taxa where Pleistocene glacial cycles have been important for diversification events. But one thing about the previous study is that it used gene trees. So we now know that this is somewhat of a problem, right? We're going to get these overestimates of divergence time. And they didn't include all extant taxa. This is because since that study, there's been 5 species added to the genus, plus we're going to delimit some more today. So our methods are, we've got 276 milk snake samples across their range, at least 2 individuals from all other Lampropeltis species, plus 2 outgroups, Arizona and Sumatera, 1 mitochondrial gene where we sequenced all the individuals for this, and 11 nuclear loci. Some of these are from the literature, some of them were developed using anonymous loci protocols for this project specifically, and it's a mixture of introns and exons, and we did a subsample based on getting good geographic coverage for sequencing the nuclear genes. To delimit species, we want to use BPP, which I've already explained a little bit about. All the trees that are used in this are from the program Beast, and we have 2 different ways we made guide trees for this. So the first was just using the Seibert gene tree and seeing if that's able to delimit species. And then the program Structurama. So this is a population inference program that assigns individuals to groups or populations based on trying to put them in Hardy-Weinberg equilibrium. One thing about Structurama is that it doesn't give you a tree. The output is just what your populations are, who's in each population. So we took those populations, ran that through Beast to get the topology to use as that guide tree. And we did this based on mitochondrial and nuclear DNA. In addition, we made a nuclear network in the program splits tree so that we could see if there's any reticulation or hybridization going on with nuclear genes between any of the potential species. So then to infer the species tree and the divergence times, we used Star Beast for this, using the newly delimited taxa we get out of BPP, 2 fossil calibrations that have log normal distributions for Lampropeltis. And we also wanted to further explore this issue of gene tree methods, so that would include concatenation and whether they're really overestimating divergence times when you're using all the same taxa. So we made this concatenated tree also in Beast, and then we also used Pybus and Harvey's gamma statistic to take a look whether that node density does in fact shift. So the results show that we've got 11 lineages of milk snake that aren't necessarily even sister to each other in that Site gene tree. And Lampropeltis alterna has the same exact or very similar haplotypes to a lineage of milk snake in the Southwest. So what am I saying? Or is alterna really secretly a milk snake? Well, the nuclear genes say no, that's not the case. So you don't really need to worry about it too much. Structurama gives us these 6 milk snake groups, plus I threw alterna in there to see what was going on. Turns out it comes out as a distinct population, so it's probably some mitochondrial introgression going on here with alterna. All of these lineages or groups were delimitable in BPP, but those Site B lineages had lower support, especially under more conservative speciation priors. So, and also there were, some of those lineages only had a couple samples in them, so not necessarily as robust as the Structurama results. We made this nuclear network. Again, I threw alterna in there to see if there was any potential gene flow going on, and we got 7 milk snakes plus a distinct Lampropeltis alterna. And ultimately, we've delimited 7 milk snake species with high support that we use in our species tree inference. So if we take a look at just very quickly how these fall out across the US, what their names are as a species, and what subspecies would be synonymized with each, we have eastern milk snakes, triangulum in the eastern US. We have gentilis in the western US. We've got Elapsoides in the southeast, annulata in eastern Mexico, Polyzona in western Mexico, Abnorma throughout Central America, and Micropholus picking up in Costa Rica and all the way down into South America. We look at the species tree, we see that we've got pretty high support for a lot of our relationships among taxa. Milksnakes aren't monophyletic yet again, there's 3 distinct groups. And most divergence does occur in the Pliocene and in the Pleistocene, not further back in time when we use the species tree approach. I'm not going to show the concatenated tree topology, but if we compare the results with the divergence times to the species tree, we find that most extant species for the species tree are originating in the Pleistocene, while most in the concatenated tree are originating in the Pliocene and Pleistocene. Additionally, the gamma statistic for the species tree says node density and speciation events are pretty much constant throughout time. There's no early burst or late burst, it's pretty even. The gamma statistic for the concatenated tree gives us that greater node density in the past. So just like that first slide I showed where it shifts from being somewhat even or early to being much later. Or, I'm sorry, being early and shifting to being later in the species tree. So if we further examine the differences between the divergence times, the concatenated tree mean dates are overall older and significantly older than the species tree. And if we take a look here at the difference in mean times, which is the branching time error here on the y-axis, and compare that to the branching times of the species tree in this linear regression, we see that as at more recent divergence times here, closer to time 0, that error between the two is much greater, and that as you go further in the past, that divergence time error is somewhat smaller. So this may be a problem a bigger problem at more recent times for detecting divergence events. So what does all of this mean? Well, we have the addition of 6 new milksnake species to Lampropeltis, and when we use a species tree approach here, we find that milksnakes aren't monophyletic within the genus, and so they've been misidentified or misclassified as being either a single species or a group of species. They're 3 distinct groups. And that when we include more species, we have more speciation events happening during the Pliocene and Pleistocene. So why do we have all these recent divergence times now? Well, we're including 21 species, not 10. And we've got sister taxa splitting at more recent time periods. So a lot of these species are newly formed, not newly formed, but new to this tree, sister taxa pairs. And so we're getting these more recent time periods during the Pleistocene for the speciation events. Also, additional studies for some of the other species in this genus might show us that there's even more recent speciation events where we've got things like Zonata or Caligaster that have wide ranges and haven't necessarily been looked at in a phylogeographic context with delimitation methods. So we may result in even more Pleistocene divergence. Additionally, we find that gene trees do in fact overestimate divergence times. So the concatenated tree gives us times that are mostly in the Miocene and Pliocene versus the Pliocene and Pleistocene for the species tree. The concatenated tree times are significantly older, especially at more recent time periods. The concatenated tree also has greater node density in the past, signaling an early burst of diversification. So we see that we're getting a different interpretation of this downstream analysis when we use a species tree gene tree rather than a species tree. And so we can maybe conclude that diversification did continue during the Pliocene and Pleistocene for Lampropeltis, and glacial cycles may in fact have contributed to diversification, or at a minimum didn't seem to slow it down. It didn't seem to have a negative effect on speciation for these guys. And so to conclude, we can say that the diversity of milksnakes and for the whole genus has been previously underestimated. Efforts to include all extant taxa are vital to estimate both topology. We find out that when we've got all these species and not one single representative, that milksnakes aren't even monophyletic. And our divergence events, if we want to properly estimate those times, we are going to need to include all our taxa, because now we have more sister taxa that are splitting at more recent time periods. And ultimately, we want to use species trees and not gene trees. Hopefully, we're getting more accurate divergence times because we're taking into account all the individual histories of each gene. And this is especially true for recent diversification. And so with that, I would like to thank everybody in my lab who's helped out with this, everybody who's donated tissues. I think half of you are probably in this audience right now, so you know who you are, and everyone whose photos I used in this talk. Thank you.
Thanks a lot. There's time for one very Question, yes.
So how do you think this will change the prices of milk snakes in the pet
Oh, in the
trade?
pet trade. I don't think people in the pet trade are going to be very happy. There aren't so many distinct subspecies now.
Okay.
Thank you.
Sara?